everyone, and welcome to today's webinar. Maximize your cell health assays data acquisition with latest tools in cancer research. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com. Now, we encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation, and we will assist you directly. I would now like to welcome our two speakers, Nicholas Chia, Technical Sales Specialist, Protein and Cell Analysis, Taiwan, Thermo Fisher Scientific, Singapore, and Edmund, Technical Sales Specialist, Protein and Cell Analysis, Asia Pacific and Japan, Thermo Fisher Scientific, Singapore. For complete biographies on our speakers, please visit the biography window at the top of your screen. Edmund, Nicholas, welcome. You may now begin your presentations.
Thank you for the introduction. In today's webinar, we will dive into the tools and reagents for measuring cell health in cancer research. So let's get started. In cancer research, scientists use different models to understand how the disease works, from basic cell cultures to animals and even human subjects. For discovery purposes, scientists often rely on 2D and 3D cell models to study sensor biology and test out new drugs. Each of these models has its pros and cons, as indicated on the slide, that researchers should consider carefully before starting an experiment. Understanding cell morphology, health, and other structural and functional aspects is crucial in cancer research. These factors help scientists dive into cancer cell metabolism, stress responses, organobiology, the cell cycle, and post-transcriptional control. In addition, they play a pivotal role in pinpointing potential therapeutic targets and developing effective treatment. We'd like to use this diagram to highlight key aspects of our discussion. By examining cell health from healthy cell reproduction through stimuli that induce various stress responses to cell death and post-mortem changes, we can better understand how normal cell functions are disrupted in cancer. And what I got here are several key points we aim to address. We have developed tools and identified needs within the research community to recognize healthy, viable cells and the activations of protective mechanisms intrinsic to these cells. By considering our cell health continuum, we can observe the effect of pre-lethal stresses such as mitochondrial toxins, drugs that affect the cytoskeleton, and cell cycle inhibitors. We can then examine the outcomes of these stresses, which include caspase activation, DNA damage, downstream loss of plasma membrane integrity, and the phagocytosis of dead cells. So typically, in cancer research, cell viability, proliferation assay, and stress assays are commonly used. So we have highlighted some of the assays that can be used on both imaging and plate reader platform, such as caspase 3.7 reagents, live date kits, and more. And cell assays can be conducted and analyzed using different methods, including microplate detections and fluorescent imaging. So in today's webinar, we will focus on two approaches where the same sample plate can be utilized for both plate reading and imaging. The dual use of the sample plate allows for comprehensive data collection, providing both quantitative measurements and visual confirmation. Additionally, using the same sample plate for both methods minimize variability and conserve valuable resources. A microplate reader typically offers three different detection modes, such as the absorbance, fluorescence, and luminescence. Each detection mode has specific applications, advantages, and requirements based on the type of assay and the properties of the samples being measured. So to achieve optimal performance for each detection mode, it is essential to select the appropriate plate type. And in this webinar, we will be focusing on the fluorescence-based cell health assay. So in this slide, I would like to introduce the Presto Blue cell viability assay. Presto Blue is a resazurin-based assay used to assess cell metabolism. As resazurin is metabolized to restorufin, the solution changes color and exhibits increased fluorescence intensity. And so this assay can be detected using both photometry and fluorometry. The protocol is simple and direct, offering stable signals for approximately 4 to 6 hours, providing researchers ample time to conduct experiments. And on this slide, I'm providing an example from this published article where researchers tested the cytotoxic effect of triptolite on MCF7 cells across different concentrations. Cell viability was evaluated with Presto Blue after treatment lasting 24, 48, or 72 hours and showed decreased cell viability following the compound treatment. After determining the effect of triptolite, the researcher investigated further into the compound's mechanism of action. They discovered that triptolite exhibit potent anti-cancer effect by targeting the estrogen receptor alpha-mediated signaling pathway. To confirm the hypothesis, the compound treatment was performed on both wild-type and ER-alpha knockdown cells. In the experiments, they found that when ER-alpha was knocked down in cells, the anti-cancer effect of triptolite became insignificant compared to its effect in wild-type cells. This confirmed the cytotoxic activity of triptolite is acted through the ER-alpha receptor. And viability is not easily defined in terms of a single physiological or morphological parameter, and relying on a single measure can generate bias in the experiment. So in lead identification and orthogonal assay approach eliminates false positive or to confirm the activity identified during the primary assay. In the hit identification process, false positive may occur due to non-reproducible activity or interference in the primary screen. The initial activity observed may be due to the test format rather than the biology of interest. So to eliminate false positive and confirm activity, an orthogonal assay is performed after the primary screen by using a different reporter or assay format to confirm the activity is directed toward the biological target of interest. And in this, I would like to share another type of cell health assay, which is cyclone proliferation assay, which can be used as part of the orthogonal approach. The assay kit comprises of two components, the cell-permanent nucleic acid dye and impermanent background suppressor. The nucleic acid stain is a cell-permanent dye that binds to DNA in mammalian cell nuclei with binding proportional to DNA content and independent of metabolic state. The background suppression dye, impermanent to live cell but able to enter compromised dead or dying cell, masks the DNA's fluorescence in these cells. Because cells are not lysed or permeabilized for the assay, and because staining does not require active metabolism, the readout provides a direct measure of cell proliferation and viability. This combination allows the assay to measure both DNA content and membrane integrity, making it useful for assessing cell viability, proliferation rate, and cytotoxicity. And the cyclone assay is sensitive and accurate with the linear readout from 50 to 50,000 cells that make cyclone ideal for high throughput screening using 96 and 384 well plate. So we can use both cyclone proliferation and Presto Blue assays on the same cell culture plate to gather different insights. So in this two-step cell viability test, we will start with the Presto Blue assay, followed by adding the cyclone direct proliferation reagent and measure the fluorescence at different wavelengths. This method allows us to assess the cell metabolism, DNA content, and membrane integrity in a single sample, giving us a comprehensive view of cell health and viability. And to perform the fluorescence-based assay, we need to use an instrument that can measure the fluorescence light. So Veriscan Alpha is one of the multimode plate reader that Thermo Fisher provide for absorbance, fluorescence, and luminescence assay. The instrument also equipped with the automatic dynamic function, which is designed to ensure the instrument detect sample reliably at a wide signal and concentration range. The instrument automatically adjusts the measurement gain for every sample on the microplate individually. and the user no longer needed to perform test run to optimize gain settings separately for each assay and plate. Additionally, the Rescan Alpha also includes essential feature for cell health assay, including an incubator and shaking capabilities. So before we dive into the demonstration, let's go over the material and methods. Prior to the cell health assay, we will expose MCF7 cell at various density to different concentration of sterosporine for about 14 hours. So you can view the plate layout on the right side of the slide. And following the compound treatment, we will introduce the assay corresponding reagent into each well and measure the result using both a microplate reader and microscope. Next, we will show you the demonstration on plate reader.
Today, I'm going to show you how to run two different types of assays on the same plate and how to easily determine the ED50 using the ScanIt software. So first, we will start by clicking the New File button in the top left corner and selecting our instrument model, which will be the Veroscan Alpha. This is the instrument we will use for detection later on. So on the left side of the screen, you will see the experiment file is divided into 5 sections. In the first section, there is a section note area where you can type in the details of your experiment. This information can later be exported to Excel or PDF for record keeping. Next, we will move to the plate layout sections. Here we define the samples on the plate. So there are different sample type labels available, such as blank, standard, control, and unknown. Defining the samples here is crucial because it allows us to use the result analysis function to plot the curve easily. As shown in the demo slide earlier, we will now set the plate layout. In our demo, we are using two different types of assay, so we need to create two sample groups for each type of assay. So remember to select the correct sample group. If there are replicates, we need to check the replicate functions and specify the number of columns and rows depending on how the user arrange the plate layout. For example, we have triplicates for our Presto Blue control from B7 to D7. So we will put 1 column and 3 rows. Next, we will define the standards. We will enter the concentration of sterosporine starting from 2 micromolar down to 0.125 micromolar. For compound screening, it is advisable to include a blank to subtract the non-specific fluorescent signal emitted by the compound, which might interfere with the readout. However, due to time constraint, we will not include the blank for this demonstration. Select the wells and drag and drop them accordingly. So we will repeat a similar process for the caspase 3-7 assay. So after setting the plate layout, we will go into the protocol section. So since we are using two types of assay detectable with different fluorescence wavelength, we need to use the area selection function before choosing the fluorescent detection mode. We will select the area specifically for Presto Blue and then insert another area selections for the caspase 3-7 assay. So under each area selection, we will insert the fluorescent detection method and choose the appropriate filter pair for each assay. So for example, we will use the orange fluorescence filter pair for the Presto Blue assay and the green fluorescence filter pair for the caspase 3-7 detections. So in the advanced parameter section, there is an automatic dynamic function that helps user to determine the gain setting automatically, as I have shared earlier. Additionally, if you scroll the page to the left, you will see a spectral viewer. This tool allows user to check the excitation and emission peak of commonly used fluorophore. So we will repeat the same process for caspase 3-7 assay. So if shaking is required before the measurement, we can add the shake function. You can adjust the shaking type, method, and speed under this function. The sequence of functions can also be rearranged simply by dragging and dropping them. Next, we will move the cursor to the bottom left of the screen to eject the plate holder and insert the plate by clicking the green triangle. Once that is done, we can now start the run by clicking the start button. The instrument will perform a pre-execution check to ensure the plate is in place. To view the measurement in real time, go to the result section. So the first readout will be for the Presto Blue assay, and then the system will switch to a different filter pair for the caspase 3-7 assay. And next, I will show you how to plot the dose response curve using ScanNet software. So first, click on the average function button to average the readouts from replicates. And then click the normalization button to generate data in relation to drug activity as a percentage compared to the control. Finally, click the dose response curve button and with just one click, the curve will be plotted. In the dose response curve function, you can choose from different fit types, both linear and non-linear model. So the median effective dose will be calculated and displayed below the graph. For this experiment, using the Presto Blue assay, the ED50 is 1.746 micromolar. So with just 3 simple clicks, you can generate a dose response curve along with the calculated ED50 easily. And next, we will do a quick analysis of the caspase 37 results. Since we are running this assay for a qualitative check to compare the control and treated samples, we will use the average number for a quick screening of caspase activation. As I have shared earlier, we have several reagents that can be used on both plate readers and imaging platforms. We can further verify the caspase 37 assay results using our fluorescent microscope to observe changes in cell morphology. And Edmund will demonstrate this in his session later on. At the end of the experiment, you can toggle to the report section to export the data to a PDF or Excel file. Here you can choose what information to include in the export. Once you click on the export button in the top left corner, the file will be generated immediately with all your selected information. So here comes to the end of my demonstration. I will now hand it over to Edmund, who will share how researchers could utilize the microscope for cell health assay. Thank you.
Okay, thank you, Nicholas, for sharing in the first part of the webinar. So as Nicholas has already mentioned, cell health assays can be read in various methods for categorization and analysis. And I'll be touching on the second part where imaging allows us to view and understand the functional and morphological changes to your cells. So in cancer research, many assays such as cell viability, live date, and cellular stress assays are commonly used. I'll be touching a bit on these three assays, where we have common ones that Nicholas has also mentioned earlier on, that we can be using it for both imaging on the microscopes and on the microplate reader. So coming back to the pathway of the cell, we will be actually touching on the first part, which is the oxidative stress. So reactive oxygen species, ROS for short, are produced by both healthy and stressed cells. So sometimes the cell loses the ability to regulate this species, or sometimes when you're actually doing drug treatment, the drug induces the overproduction of this reactive oxygen species. So in both cases, they are actually a good mechanism for representing a classical tool for identifying the mechanism of action in drug treatment, or understanding how a gene has altered the metabolism of a cell, particularly with the mitochondrial phenotyping. So researchers have already been using our classical dyes to study reactive oxygen species for many years. However, the reduced fluorescent dyes suffer from the inability to use them in complete media, and they typically photobleach very quickly. So with the CellRox dyes, deep red dyes that we have, they are currently also available in the green and orange forms. They recognize the reactive oxygen species, and they can be fixed and are very photostable. So these actually work on both live and dead cells, so you can visualize the activity of the dye. Okay, in the multiplex example here, so the CellRox green dye, a reactive oxygen dye, is a nuclear binding dye that binds to the DNA upon oxidation. So the dye is dark, as you've seen earlier on, and it oxidizes and then it moves to the nucleus and binds to DNA. Then it produces a very bright and localized green signal in response to the reactive oxygen species. So as you can see in the mitochondrial signal, it becomes dimmer along the way with the drug treatment, and the ROS signal is appearing with time. So the next portion that we'll be talking about is the activation of caspase enzymes to induce cell death. So we all understand that programmed cell death is a well-known phenomenon, and the main executor of this programmed cell death is actually the caspase 37. So caspase 37 induces cell death by cleaving cells at specific site in their protein sequence. So they actually recognize the DEBD target sequence. By using the DEBD target sequence, a binding DNA dye is fused to it. This is similar to the CellRox dye that we shared earlier on. Then it only binds upon oxidation. This caspase dye actually binds after cleavage by caspase. At rest, the dye is dark and nothing is seen. But when you induce caspase, in this case, with activation of sterosporine or another type of toxic drug, right, the dye is cleaved away from the DEBD and can translocate to the nucleus and produce a fluorogenic signal. So there are some advantages here. You don't have to wash the cells or remove the dye or the medium for this to work. The sample can also be fixed and counted after polymerization, and it can stain with other dyes. And of course, you can get a lot of amplification of the signal. Every caspase enzyme remains active and it keeps cleaving the dye as long as it's around. So in the video that you've just seen, the mitochondrial potential in red is actually disappearing, and the activation of the caspase in green is taking place. And you will notice that in this time lapse, it shows how the caspase 37 actually works. So now we also have a GFP compatible caspase 37 dye. So it operates in the far red channel, and it can also now be multiplexed with your green fluorescent proteins or deep red dyes for studying cell death and nuclear staining in the blue channel. This actually gives you more multiplexing options and the flexibility in the design of the experiment. Okay, so for live imaging, we can also perform viability assays with our live and dead kit. So live and dead kit is actually a very quick and easy two-color viability assay. So it is actually based on the membrane integrity and esterase activity in the cell. So healthy living cells will be stained very brightly with the calcium AM dye. It will appear in green as it interacts with the intercellular esterase. So for dead cells, they actually have compromised plasma membranes. It will allow the actinium homodimer 1 to actually enter the cell and bind to DNA. This process emits the red fluorescent signal, and this kit will allow you to study the live dead population of your cells. So this kit is actually very easy to use. It can be added to your cells and incubated at room temperature for about 30 to 45 minutes. With this viability kit, right, you can also image the cells on the microscope or read it on the microplate reader. So here I would like to share a bit on the typical workflow for imaging. First of all, you'll be preparing your cancer cells with an optimized protocol. A simple microscope such as the EVOS XL Core or the M3000 will actually help you in monitoring your cells. Then subsequently, you'll be treating your cells with your drugs or chemical compounds and staining them subsequently. So a fluorescence microscope, for example here, the EVOS M5000 or the M3000 can be used to actually check your labeling expression. And downstream, you can actually do more of the analysis using flow cytometry or automated microscopy and even high content screening to get more data for understanding the population of the cells. Now, I would like to share a bit more on the EVOS microscope family portfolio. So as I mentioned earlier on, there are actually two new models which I would like to call out. It's actually the EVOS M3000, which will be launching next month in August. And for today's webinar, we'll be talking on the EVOS M5000 with stage view. So with the EVOS M5000 stage view, right, we can actually have that unique intelligent tracking function to allow you to maximize your ability to do your experiments. Now I'll be sharing a bit more on the EVOS M5000 microscope. It's actually an all-in-one compact, semi-automated microscope that allows you to maximize your cell health assay data acquisition. So you'll be able to acquire up to 4 different fluorescent staining on your cells, as the automated multiplexing helps you to acquire those channels, plus a phase contrast transmitter light image with the simplicity of a click of a button. And with the system, you actually have autofocus and time-lapse functions. So it actually allows you to add more dimensions to your experiment. So if you're working with even 3D cell cultures or 3D spheroids or tumoroids, the Z-stacking function on the M5000 allows you to acquire 3D data and actually reconstruct them in analysis software such as our Celeste software. So the EVOS M5000 also has onboard cell counting and confluency measurements, so you can actually do some basic cell count analysis on the system. And on top of that, you can actually add the EVOS onstage incubator for light cell imaging on the system. It enables precise control of temperature, humidity, and 3 different types of gases for time-lapse imaging of your live cells under both physiological and non-physiological conditions, such as hypoxia. So all these environment settings, right, and the imaging acquisition parameters are all seamlessly integrated into the EVOS software. So it's very easy for the user to actually learn and operate the unit. And on top of that, we actually have the ability to put up to 4 fluorescent LED light cubes. So each EVOS LED light cube is actually specific for a certain region staining. So it also allows you to minimize the crosstalk between channels. Here are 5 of the more common, I would say, LED light tubes that we are offering to our users. So they are able to cover most of the, I would say, common dyes in the market. And here are the 3 that we mentioned earlier on for apoptosis, oxidative stress, and even variability. So all these 3 assays can actually be done on the EVOS microscope with the appropriate LED light cubes. So now, I'll be touching a bit more on how the stage view or this function actually helps you in your experiment. Imagine now you're working with cells and you want to monitor your cells during a treatment over a certain period of time. There are a lot of cells which could be inside the well, and you wish to monitor them over time. So once you actually identify the cells among this sea of cells in this well, you will be wondering, how are you able to come back to the same location to find these cells? So imagine, for example, here, this group of cells, and you are identifying this red spot. This is the cell of interest that you want. But this is going to be quite challenging for you to come back to the same position to look for them. Imagine that this is only one location in the well. How are we actually going to find the other positions in that well and imagine across the whole multi-well plate? So let me share with you more on how the EVOS M5000 with stage view allows you to do this with ease. So first of all, the unique location tracking function on the EVOS M5000 is actually due to the integrated XY sensors on the stage. This allows the stage to identify the location where the sample is. And of course, with different vessel holders, we can actually use them to suit the type of vessels that you're working with. For example, multi-well plates, general slides, 35 dishes, or any other type of vessels that you are planning to work on. So the stage view function or on the M5000 allows you to return to these locations to find your cells. So here is an overview of how the stage view software interface looks like. You'll notice on the right bottom corner, this is the stage view window, which you can see in the software. So as mentioned, there are actually different type of vessel holders which you can use, and this will be matched in the stage view vessel maps. So we do have vessel maps for the standard 6, 12, 24, 48, 96, and 384 well plates. We also have it for standard slides, 35 Petri dishes, and if you're using something different, we have the blank map option that allows you to use it on the EVOS M5000. So some cases where you can imagine to use this feature, right, is that you are looking for long-term self-growing monitoring. So you do not want to put the cells on the microscope for a long period of time. You can take it out, image it, and put it back in the incubator and come back at a later time to re-image the same location. And of course, you can also mark areas of interest on histology slides. So you can also find suitable candidate cells for physiological measurements. And of course, if you are doing a teaching lab, it allows you to guide the students to view some biological samples when they are actually looking at the whole well plate or certain slides. So now, in the EVOS stage view software, once you have selected the vessel map, right, and you have placed the vessel on the stage, you can actually identify the cell of interest, and then on the plus icon here, you can actually add multiple pins and relocate them. So here, example that I've added a pin here at well A3. You can see that you can add multiple multiple pins on different wells, and you can come back to them. So for example, here I've placed 99 pins on this 96 well plate map. So we can look for the location of specific pins or the cell of interest, and you can relocate them accordingly. You notice that once you click onto the find pin map, right, it gives you the overview of the 96 well plate, and the pin that you are of interest now will actually appear in orange. Then I will actually be using pin number 4 as an example in well H8. So you see that once we actually move the stage towards the location of pin number 4, the orange icon, or rather the orange pin, will actually change into green. This means that you have come to the vicinity of this pin, which you will be able to relocate it much more easily. Similarly to how a 35 Petri dish is used, we actually have the vessel map on the stage view function. But you'll be wondering, how are we able to use it on the 35 dish? So first of all, we will always advise our customers to actually draw a marking on the bottom of the Petri dish. Do remember that it's not on the cover, because the cover is always freely rotatable. So once you have that marking on the Petri dish, then you'll be able to align it with the vessel holder. So with this, you can actually add pins on the map and relocate your cells accordingly. Similarly, for standard slides, you will be placing them with the label on the right-hand side, and you can actually then drop pins to find the locations of the structure of the cells or on the tissue that you are interested in. And last but not least, the blank map option gives you an ability to deal with different type of vessels apart from the three that I've just shared. So it allows you to also drop different pins at different locations and come back to them to find your cells. Here is an example that we have done, that we are tracking the U2SO cells over a period of 12 hours. You'll see that we are able to identify and relocate the same location of the cells over a time course of 12 hours at different time periods. So this allows you to actually find the cells that are of interest to study them with the stage view feature, and then you can monitor them across different time points and see how your drug treatment actually takes place for your cells. So now I'll be doing a live demo of how the stage view feature works on the EVOS M5000. Here we are looking at the user interface of the new EVOS M5000 microscope with stage view. So as you can see here, you are actually able to multiplex up to 4 different fluorescent channels with your transmitter light phase contrast images. So this gives you a lot of flexibility in terms of designing your experiments. And with the new intelligent tracking feature on the M5000, the stage view allows you to track your cells over time at the same position over and over again. Now we'll be doing the calibration of the M5000 stage. Here in the stage view window, you can see they can use 4 times or 10 times objectives. We'll be using the 10 times objectives to do the calibration. Once that's chosen, we'll click on next. Then we'll now move the stage with the coaxial drive towards yourself and then to the right hand side of the microscope body. So once that is done, we can actually click next and then do the calibration of the origin. So now I'll be placing the vessel holder for the multi-well plates onto the stage, and we'll try to look for the position of the pinhole on the vessel holder to align it with respect to the stage. So you can actually manually see on the stage itself to find the location where you are nearer to the pinhole with the alignment of the light. Here we are. So you can see that I'm trying to align the crosshair to the center of the pinhole. Okay, so it's around in the center now. We can now come here and click on Set Origin and choose the vessel that we want to use. So here is the list of options that you have. I'll be using the same 96 well plate that Nicholas was using on the microplate reader earlier on. So here we'll be choosing 96 well, and then we'll be proceeding to use the vessel map. So let's click on continue now, and then you will see that the 96 well plate vessel map is now shown in the stage view window. We can start using the stage view feature. I'll be placing the 96 well plate onto the stage now. Once that's done, you can start moving the stage, and here you can see in the stage view window, right? The orange crosshair will start to move according to the position of the stage which you are moving to. It will align with the well which you are planning to go to. You can see here I'm going to well E5. And the cells here are treated with stereosporine, and we have also stained them with the caspase 37. So let me first try to focus and find the cells. Okay, now let's move to the center of the well. Where you can see here we have a group of cells that are already undergoing apoptosis. We will now confirm this by checking on the GFP channel and looking at the caspase 37 signal. So let me go to the GFP channel here, and let's see. So definitely you can see that the green signal is coming from the caspase 37 staining, and the cells are going through apoptosis. So I'll pull up the crosshair here so that we can use the stage view function to look for specific locations. Probably this one here, a standalone cell that's going through apoptosis. Let me add one pin here. You can actually rename the pin, but we will use the conventional name here as for convenience. I'm looking for a second position, a second location here. How about this? This cell here with another cell that's going through apoptosis on its northeast corner. This will help us to identify this easier later. Now let me add this to the list. So this will be our second pin. And now probably a third one that's at the corner of the well. Let's try to find one. I see something interesting here. So probably let's choose this cell. It's going to be easier to identify it and add a third pin here. So now look at the find map button. You can see that we have added 3 pins into the list. So we are looking at pin 1 now. I'm moving the stage, and you can see that once the stage is near the location of the pin or that position of the cell that we've identified, it changes from orange to green. Here you go. Now we can go back to the stage and look for that particular cell. Remember, it's that cell that's standalone. Yeah, this is the one. Now we'll look at the find pin map again. I'll just look at pin 2 now. Pin 2 is the more unique one. Remember that we have another cell that's on its northeast corner. So here we go. We are in the vicinity now. Let us check the live view. Yes, here it is. So we can actually identify this particular location that we've added on the vessel map. So this is our pin 2. And now let's look at pin number 3, the one at the corner of the well. I'm moving my stage to that location, and you can see it's turned green. Now let's look at the live view. Here it is. And if we were to check on the map again, you can see that if you are at the exact position, it will give you a 00 delta that gives you the identification of the exact position. So you can fine tune it to find the exact location. Okay, so now we have done this for a single well. Let us try it for a different well. We are going to go to well F5. Let me just add one location. So you can see the cells here. Let's focus the stage and find one position to try this out. Let me use the green channel. Okay, this one looks good. A single cell that's easier for identification. Let's add this to the map. So this corresponds to this spot. I will now shift the stage away, and we will look for this position again. So now pin 4, I'll move the stage back. Let's get close to the pin. And once we are near it, see it turns green again. So let's check the cell. Yeah, this is the one. Let me just place the cursor on it. There we go. Delta is 00, the exact position of the pin that we added for the well. So let's say if you want to work with the whole well, you can actually add more pins. So at the vessel map location here, you can add the pins according to the location of the cells. You can also rename them so that it's easier for you to identify the locations. One more thing that I want to share in this demo is that we can actually do the automatic multiplexing of the channels of interest. So here we have phase contrast and the GFP channel for the caspase 37. So on this M5000 microscope, right, it allows you to automatically capture these channels. So you can see here, I have just acquired them both, and you can quickly identify where are the cells that are undergoing apoptosis. You can see these signals exhibiting in green on these cells which are going through that process. So this allows you to track your drug treatment uh, over a time period of, over a time course, so you can actually come back to the same locations and monitor them accordingly. So this is all for the live demo, and we'll go back to the presentation. Okay, I hope the demo was actually interesting for you. So now we've come to a bit on the resources that's available for you to go to. So here's an overview of the resources that we have on our websites, which you can actually read up on for cancer research, for cell analysis learning center, and of course, the imaging basics for microscopy. There's also one thing that I would like to share is that we actually have a staining cell staining simulator tool. This allows you to choose the different reagents or dyes to stain the different structures of the cell, and you can play around with this tool to choose the optimal, I would say, fluorescent stainings to actually work together in your experiment. And last but not least, for the two models which we have actually shared today, the EVOS M5000 and the Bioscan Lux, we also have included the two links here, so you can actually read up more on them. And in conclusion, there are actually various assays that are available for everyone to actually check and monitor your cells. Some of the applications, which include like long-term tracking of cells, cell migration, 3D spheroids or tumoroid formation monitoring, and even wound healings are actually suitable for imaging here. You can also multiplex your experiment with the EVOS M5000 using the stage view feature. So you can actually find your cells over and over again, and you can acquire both qualitative and quantitative data. Cell health assays will also still remain as one of the key assessment parameters in all cancer research. So using these non-destructive assays, right, such as your cell trackers, your mito trackers, your Presto Blue, and etc. Reagents, right, you can actually multiplex and use multiple assays to maximize the data acquired from your cells. So this actually helps you to increase your cell data integrity, and of course, you can save on your cell resources. The Bioscan ELF actually provides accurate quantitative readouts using both, absorbance, luminescence, and fluorescent modes. So all on one single system. And for the EVOS M5000 with stage view, it allows you to do multiplexing and come back to the same position of the cell so that you can actually track them and monitor them along the way. So this is the end of our webinar. So we hope you have enjoyed this session. We'll be open up the floor for any questions. Thank you.
Nicholas, Edmund, thank you so much for that excellent presentation. It is now time for our live Q&A portion of our webinar. Now, to our audience, if you haven't submitted any questions yet, please do so now. Just type them into the Ask a Question box, click Submit, and we will answer as many of your questions as we have time for. Okay, so we're going to just dive right in here. Nicholas, Edmund, our first question. For plate readers, how many plates and plate types can it accommodate? And what is the reading speed?
So for Alpha, you can only read one plate at a time. So it can accommodate multiple plate format, so such as 6 to 48 well, 96 well, and 3F4 well. So the reading speed for ALF in absorbent mode is less than 10 seconds for 96 well, and in fluorescence mode is less than 20 seconds.
Very good, thank you. Our next question. Could you please share more about the microplate reader software? Does it allow for pre-programmed and custom protocols? And what kind of analysis is offered?
So in scanning software, we have like a club library that actually loaded with like different pre-programmed protocol that are commonly used, such as the MTT assay, Presto Blue, DNA quantification, protein quantification, and more. So and also we can also customize the protocol because we have multiple functions on the software, which allows the user to select and drag and drop onto the protocol, and depending on their experiment design. Okay. So other than that, the software also provides like robust result analysis function. So such as like blank subtractions, normalizations, like plotting of standard curve, dose response curve, and more. So these allow user to perform like analysis directly on the software itself. So eliminating the need to manually input the formula on Excel. Yeah.
Very good. Thank you. Our next question. Is the microplate reader capable of measuring nanobread assay?
Yes. So for ELF, it is possible to measure nanobread on the system. So we actually have the filter set that is catered for the nanobread assay. So, but both filter need to be installed on the emission filter view. Okay. So unlike the traditional setup. So this configuration is designed for the luminometric nanobread assay, which do not involve like excitation. So instead, it measures the bread donor and acceptor signal using two distinct filters.
Thank you. Next question, and we have so many great questions coming in here. What is the maximum number of pins that we can set on each map?
I think I'll take that. So for the M5000 stage view, right, we have been able to set up quite a number of pins. So in the presentation you saw earlier on, I've added almost up to 100 pins in that map. So logically speaking, there is no limit as of now that we are aware of. So you can add up to probably a couple of thousand. So depending on your applications, hopefully you do not need to identify so many locations in the 96 well plate. That will be quite a lot of work to be done.
Excellent. And we have time for just a few more questions. Is there any impact on location accuracy when objective fluorescent channel changes?
Okay, so for the stage view feature, right, the pins are actually identified to the location of the stage. So in the XY sensors, it identifies that particular location. So by changing the objective turret, choosing different objectives, it does not move the location away. So you can definitely be sure that once you change the objective magnification, you'll still be able to find the same location in the stage map.
Thank you, Edmund. And gentlemen, we're going to have to wrap with this final question. And I'm going to go with this one because we've had it come in a few times. Is there any upgrade trade-in program as I own an M5000? Could you give us some details on that?
Okay. So thank you for the interest. As of now, there is no M5000 trade-in program, but definitely if you're interested in the new M5000 with stage view, do approach your local sales reps or your local sales team for more inquiries, so they will be able to address that accordingly.
Very good. Thank you, gentlemen. And I'm going to make a comment to the audience here. We did show some resources, so I have put those resources back on the screen. So if any of our audience members would like to click on those links and check out those resources, you can also find some on the far left of your screen. But gentlemen, before we wrap, do you have any final comments for our audience?
So if you're interested in the system, please feel free to contact your local sales representative for demo. Yeah.
Very good. Well, gentlemen, thank you both so much for your informative presentations and for your important contributions to cancer drug discovery. We also want to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. And before we go, I do want to thank our audience again also for joining us today and for their interesting questions. The questions that we did not have time to answer today and those additionally submitted during the on-demand period, they will be addressed by our speakers via the email address you provided at the time of registration. Today's webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your friends and colleagues who may have missed today's live event. Friends, thanks for joining us. Until next time, have a great night, day, and morning. Bye-bye.
Bye.
Thank you. Bye-bye.